here's the overall schedule. You should have this on a printout. So I think maybe before we get started, I know this is always maybe my least favorite part of sessions, but maybe we should just have everyone do a brief introduction, your name, where you're from, and what type of work you do, what's your background. So I'll start. I kind of started as a graduate student working with electron tomography, so sort of using computational techniques to reconstruct volumes with a much finer resolution than what we're discussing here. And then about halfway through my PhD, I transitioned into looking at serial block face volumes, which are gigantic data sets that cover a large range, but with a somewhat coarser resolution. And then quickly found out that segmentation was a huge problem. Getting quantitative results out of those data sets was extremely difficult. And then I kind of went down the wormhole of automatic segmentation and developed a lot of what I'll be introducing here. And a lot of this we're still working on. So maybe, do you want to start, Horn?
My name is Horn. I'm from the South Institute, and I'm a collaborator with Mark Harrison. And then I started a collaboration with Mark viral protein, which is a little polymer inside the cell. And then recently I've been working on looking at construction inside the cell. And mostly I'm working with electron tomography data. And then I'm working with coffee. And I would just say segmentation is a big important part of computation. So we have this something we give a
As Alex said, my name is Chris Churis. I'm a lead software developer on Slash, which is the portal you guys are going to see in a bit. I love working with high-performance computing. EM data like this is segmentation. It's huge demands.
My name is Shayna Watson. I'm a PhD student at the University of South Carolina, and I decided to come to the workshop because I've been using the fitting mode and continuity to determine college and fiber angles.
I'm Sue Wessler. I'm a faculty in biomedical engineering at the University of South Carolina in the School of Medicine. My main interests are vascular biomechanics, in particular related to atherosclerosis and plaque rupture, as well as aneurysm development. And we do a lot of reconstructions from biomedical images, such as CT scans, and recently I started to do some work with 50% as well, which is one of the reasons I'm here.
My name is Teresa Lai. I'm a PhD student at Columbia University. I'm concerned with generating Cardiac models from OCT wires. And I've also been using continuity to do electrophysiology simulations.
Francisco. I'm a biology student, recently graduated, and I'm volunteering for Alex, and I'm doing segmentation.
Okay.
I'm Robert Wallstrom. I'll be here most of the time. I work as the executive director for the MBCR. So we have up to maybe more technical issues as well at the conference. And my research is actually at a much smaller scale than what we talked about this entire conference. I do protein conformational ensembles and looking at how proteins function in the atomic scale. And I work for room 8 in our lab to research so welcome very much. You've got emails from me.
Okay, great. Thanks, everyone. So before I get started, just to get an idea of everyone's background, who has iMod installed on their laptop? Okay, 2. So we'll need to budget some time for that. Okay, that goes. And who has a background in microscopy? When I say things like serial block face, does everyone? So I'll go over. Okay, so I'll start with a intro from the beginning. So without further ado, we'll get to Cabbage, which is, I know, why you're all here. This is my dummy example for explaining the imaging modality that we'll be discussing for the rest of today. So you can imagine, everyone knows when you cut into a cabbage, you see all these weird sort of folds and crenulations. So what if you wanted to reconstruct what the entire folding pattern in the inside of that cabbage was? Well, one way to do that, as you see in this animation here, would be to cut through it at varying thicknesses. Let's say 1.5 inch. This guy's not a very good chef. He's making big cuts. So you can see there we have a bunch of slices we've collected through this cabbage, and at 1.5 inch increments. And of course, we could then segment those like a coloring book and then reconstruct them in 3D using some of the approaches we'll be showing. Of course, there are a lot of issues with taking this approach to reconstructing those folding patterns, a couple of which I'll go through here. The first is that, let's say we're just taking these pictures using your iPhone or something, rather than a microscope, because they're pieces of cabbage. You might have one image in the middle, as you can see, that second from the bottom, that's out of focus or blurry, and obviously it's going to be much harder to reconstruct the folds there. As you can see, you can't really make them out. Since it's a biological tissue, it might shrink, it might expand, you might lose part of it as you go from slicing to imaging to reconstructing. You might miss one altogether. It might fall on the ground, or your dog eats it, or something, and it's gone. And then obviously that's a big issue because you have no way of filling in, reliably filling in that missing data. And the last is you might not be able to put them back together in the right way with the right overlap. They might be misaligned either translationally, or you might get them in the missing order through the thickness. So the reason I started with that was to sort of highlight what some of the common issues with the historical method of doing this are, which is serial section transmission electron microscopy. So without getting too much into the microscopy, because that's not really the focus of the session, essentially what's going on here is you take a rosin embedded block of your tissue that's been prepared for EM, stained with heavy metals. And then traditionally the way you would do this is you would go take that block to what's called an ultramicrotome, which cuts really controlled sections of a known thickness, say 80 nanometers. So you can plane off sections of 80 nanometers and then collect them on EM grids like you see here. So maybe on one grid you can get 3 to 5 sections of 80 nanometer increments throughout the depth of the tissue. Then you take those grids to the TEM, image each one by one. So say we were interested in a cell in particular in that block of tissue, you'd want to image that. So you'd have to go to the microscope, put in your grid, find that section, that area you want, image it, do that for each section, take out the film, develop it, or if you have a CCD camera, obviously that's a little bit easier. But historically, this is what people did, because before CCD cameras were around, they would develop film in a dark room and then digitize them. So you can imagine taking one grid with 80 nanometer thick sections isn't going to get you a whole lot. It's certainly not going to get you through a whole cell or it might not even get you through a whole mitochondrion or organelle. So what people did, and I've put a couple of the seminal studies from back in the day using this technique, is they would do this thousands of times. So you would sit there, manually collect, in the example of the C. elegans connectome study, 8,000 sections, in the latter one, 10,000 sections, doing this all manually. So sitting at a microtome, cutting them, and then you imagine you have thousands of these grids still, and then inserting them into microscope imaging, developing the film. And of course, back then when this was done, computers were either not around or just really developing, so we didn't have a way to store all of this data. So what researchers would do is actually print these out and then trace them like a coloring book around what they were interested in. Maybe cut them out and build a 3D model and use some sort of math to deduce morphologies, volume, surface area from that. So this takes us to 2004 is when this technique of serial block face SEM was developed. So this is using an SEM instead of a TEM. So instead of getting a transmitted image, we're getting an image of electrons that are that basically come off of the surface as it interacts with the electron beam that scans it. So what this does is it automates pretty much the entire process that I just described to you. So instead of having the ultramicrotome at a bench and having to have someone manually cut all the sections, the microtome is inside of the chamber of the microscope doing the cutting for you. And the imaging apparatus is obviously inside of the microscope too. So this is, you can imagine a cycle of cut, image, and you'll see that illustrated in this video here, hopefully. Well, let me bring it over and play it. It's a YouTube video. It's not playing, is it? I always take pride in having all my videos work in presentations, too. Well, anyways, I don't want to waste time, so let's, the URL's there. You can see it illustrated in this left image here. It's basically a cycle of cutting, imaging, and then we end up with a stack of images for basically as long as you want to run the microscope. So we can easily collect those, thousands of sections in days to weeks time in an automated process that isn't dependent on a highly skilled microscopist to sit there and do the sectioning and do the imaging. This is just addressing some of the pros and cons of both techniques. Obviously, the cons of the serial section TEM technique, or it's very labor intensive. You can lose sections, like I showed in that Cabbage example. Section loss isn't so much of an issue with the serial block face because the block is in there in a controlled position and it never is removed until the imaging is done. The other major advantage is that the images come out pretty much completely aligned with one another because the block is just staying in the same translational plane and then just moving up in Z as it's cut. So ideally, your images are coming off already aligned. We do a few alignment steps to really fine-tune it, but at a really gross level, they come off perfectly aligned. And we're able to get, using some of the staining techniques that were developed in our lab, we're able to get images on the SEM that pretty much, unless you're a really, really highly trained microscopist, you won't be able to tell the difference between them and something that's coming off of a TEM. So we can still achieve, pretty good resolutions. We can get pixel sizes around, 3 nanometers laterally, and the cutting thickness can go down to 15 nanometers. Typically, we use, say, 30 to 60 nanometers step size in Z. So the point of introducing this was, of course, to show everyone, give everyone an idea of what the images we'll be looking at subsequently are, and then to kind of reiterate the point that a lot of the problems with collecting these data sets are mostly solved at this point. We can get big volumetric EM data sets automatically and pretty painlessly. and that the onus is pretty much now on what do we do with them. So at our lab, we collect terabytes of data a week. So one of these data sets, say we collect 1,500 slices in Z, that's already over a terabyte of image data. So how do we analyze that? How do we get any useful data out of that instead of just having a nice stack of images to look at? So jumping forward here, on the left is sort of a low magnification view of what the block face looks like inside the chamber of the SEM. So in that red box there is about the size of the volume that we acquire. Using our camera, we get 32,000 by 24,000 pixels, a raster size in XY, which you see illustrated here. Again, resolution's about 4 pixels in the XY plane and about 30 in Z. Again, that can go a little lower, it can go higher, it depends on what you need to get out of the data set. So here is just an animation, really rough animation playing through the slices of the stack. And what you'll see here is it's going to zoom in, showing kind of the multi-scale nature here. Of course, we have in that one plane, we can see about 60 to 80 cell nuclei. Zooming in, we still have the resolution to discern individual organelles. You can see here what I've pointed out, nuclei, nucleoli, mitochondria, lysosomes, even synaptic vesicles there in that. Let's see if we can see some. There's synaptic vesicles around, ER, Golgi apparatus. It's all stained and it's all visible and it's all quantifiable. So this, again, comes back to, well, how are we going to do this? Obviously, it's huge. Another question is, well, what kind of questions can we answer having this type of data? So you can imagine having some sort of disease model that you know affects mitochondrial ultrastructure. Maybe there's less mitochondrial volume. Mitochondria are more or less branched. It interrupts some of the fission-fusion machinery of mitochondria. So what you can see here is we've segmented entire neurons in the plane of that data set and then automatically reconstructed all of the organelles inside using the tools that will be introduced today. So there's a nucleus in yellow. All the green guys are mitochondria that were automatically reconstructed. Nucleoli inside of the nucleus. And this is only shown in one cell for clarity, but you can imagine each of those cells filled in with all of that data. And all those are quantifiable mesh structures. We can get volume, we can get surface area, we can get number, we can analyze clustering, branching patterns. All of those things can be quantified from these data once we have those meshes available to us. So just to illustrate the manual segmentation process with a simple example of a sphere rather than a cabbage, because I didn't want to actually trace all the folds in the cabbage. You can imagine cutting sections off of this sphere, imaging them using, again, maybe your iPhone camera in this case. And then what a user would do is sit there pretty much like a coloring book with a mouse and draw in each of those circular cross sections, yielding what are called contours there. And then using a meshing algorithm, like one of the crude ones I'll introduce today in iMod, or later in the week you'll learn about more advanced meshing algorithms, we can reconstruct that sphere like so. So you might be wondering, why do we need all of this data? There are techniques like stereology for getting things like volume fraction of mitochondria that don't need this much data, that don't need terabytes of image data to get to. So here, as an example, is one slice, a very small tile of a slice from a serial block face data set boxed out. And those pink overlays there are a mitochondrion. So you can imagine if you looked at this data set without any 3D context, you would just naively assume that those were two different mitochondria because they're spatially separate from one another. again, if we just took this single slice, you would assume that that's one mitochondrion because it's now connected. So without all of those intermediate slices showing us how that morphology evolves across the thickness of the tissue, we wouldn't be able to know that that mitochondrion is actually a Y-shaped branched mitochondria. We'd either assume that it's, you know, an ellipsoid shape or two different ellipsoids, for example. So without all of that data filled in between, we really can't quantify things like branching patterns or distributions easily. Of course, as you saw before, that mitochondrion is only one out of hundreds or thousands in the data set in that single cell. So you can imagine already how long it would take to sit there just on that, however many slices that is, and trace the cross-sections of that mitochondria fully. So I did imagine how long that would take because I didn't want to spend all my time doing this. So I did some back-of-the-envelope calculations on, well, how long would it take me, given my speed that I timed with a stopwatch, to reconstruct every single mitochondria in this volume, the one I showed you, the 32K by 24K by 1300 slices volume. Well, it would take me sitting there for 3 years, nonstop, 24/7, 365 days a year, tracing to do it in just one data set, or analogously 3 people working around the clock for a year. And that's just one data set. So you can imagine if you have different conditions, a wild type versus a knockout, or you wanted to have a higher sample size, you're just multiplying that every time. Of course, this is only a, say, 500,000 micron cubed volume. If you wanted to do even a small brain region like the suprachiasmatic nucleus, which is only about 10,000 neurons, which is very, very small compared to a lot, it would take 250 people working around the year. And then obviously it scales there. Doing a whole mouse brain would take the population of a large city, and doing a whole human brain would take most of the world working around the clock for an entire year. So obviously that's not feasible, unfortunately. And this was something that I tried at the outset, was not employing the whole world, but recruiting a number of people to help trace. And it's very difficult because you have varying schedules, you have varying degrees of competency, you have individual user biases that you have to account for. There's a lot of problems with even taking this approach at a small scale. So this led me to look and see, well, can we do this automatically? You know, is there a way to automatically segment these data sets? As you see here in this comic, that's not exactly an easy thing to do. It's something that we have methods available, but of course none of them are perfect. So the goal is to get it as perfect as possible so that we can trust the data that are coming off without really having to do too much manual work. The idea is to just offload as much manual work on the machine as we can and limit the amount of time the researcher needs to spend doing this so they can do more experiments, of course, right? So in the world of automatic segmentation algorithms, there are basically two genres, let's say, supervised versus unsupervised. Unsupervised are purely automatic. You give it the image, you write the code, and it gives you the results. You don't need to do any tracing. You don't need to tell it what it's looking for outside of what's written in your code. Examples of this are simple thresholding, where you just set, say, a pixel intensity value, and you put everything above that to a white pixel corresponding to a true hit, and you put everything below to a black pixel. I'm sure most people have done this to some level. That's the most basic method, and then from there we can do more advanced things, contour evolutions, watersheds, things like that. The problem with these are that, as I'll show in the coming slides, EM data are very complex. It's very hard to just say this grayscale range corresponds to just mitochondria and that's it. That's never going to happen with EM data. It might happen with light microscopic data, but certainly not EM data. So that the way that people circumvent this is then by installing in the code assumptions specific to certain organelles, like a mitochondrion is only this size, it's only, roughly has this sort of an outline, but as you saw from the images I showed you before, a lot of those assumptions aren't always valid because we have highly branched mitochondria, we have long ones, we have short ones. So it's very difficult to just impose a set of universal rules for this type of a process. On the other hand, there's supervised segmentation, which is pretty much the gold standard that most people use for managing large data sets. The drawback of this, of course, is that it requires human interaction. You have to train a machine learning algorithm what it's looking for. You have to tell it what a mitochondrion looks like. You have to tell it what a nucleus looks like, whatever you're interested in tracing. And then from whatever you feed it, it then extracts the features that it needs, such as, for example, image histograms, the spread of pixel intensities, various filters applied, edge filters, Gaussian filters. What does the data look like when you do that? And you can apply as many filters as you want, and it can learn from those, okay, this is what a mitochondrion looks like when I apply all of these operations to it. And then it will go through a naive image, an unlabeled image, go through on a pixel-by-pixel basis and assign probabilities of how certain it is that those pixels belong to, say, a mitochondrion or not, based on what you've taught it. So the sort of mutually shared drawbacks are that they're both time-intensive, and luckily at this stage, most of the time-intensive nature of it is offloaded on a machine rather than a person. So to train the algorithm what you want it to segment might take a new user a day to generate that much training data. It might take me, who's been doing this for years, maybe a few hours to generate that amount of data. So if you compare that to years for one person or ridiculous amounts of time for whole brains or even multiple data sets, it's pretty clear that this is a much more favorable approach to the process. So as I promised, here's an example where I've taken in just a little tile from a whole serial block-based data set, and I'm going to try to threshold it. This is pretty much everyone's first approach at trying image segmentation automatically. You can see on the right already what the problem is. That histogram corresponds to that image. It covers almost the entire range of grayscale pixel values. So we're going to have a really hard time just delineating a set of pixel intensities that correspond to individual organelles. The sort of protocol or workflow for this is that you first blur the image. If you applied any sort of pixel intensity without first blurring the image, you would get unbelievably terrible results. Then we set either just one level, like I said, where everything above it goes to segmented, everything below it doesn't, or you can set sort of an upper and lower threshold, so you're just keeping the intensities between those thresholds, which is what I'll show here. And then, of course, you're just going through on a pixel-by-pixel basis and saying, does it match this criteria or not? So here, for example, and I tried really hard. This was my best attempt at segmenting the mitochondria from here just using a thresholding approach. So I blurred the image. I selected what looked like the best upper and lower threshold levels that you can see highlighted there in that yellow box on the histogram. So everything inside of that box becomes a red pixel here, and everything outside of it is not. So as you can see, by flipping between these here, for example, those are 3 mitochondria. You can see it got a couple of them really well. It did one really poorly, but probably the biggest issue is all of the false positives we have here. So things that aren't mitochondria that were actually marked as mitochondria. So you could obviously never trust this without doing a lot of post-processing and, like I said, making those assumptions about what the mitochondria looks like in terms of shape and maybe trying to use some other algorithms to make it look better. But by the time you do that, you're going to spend a whole lot of time developing something that may very well only work for your exact data set and might not be scalable to other data sets or anatomical regions. So moving on, what I want to spend the next few slides going over is the process, just sort of putting out a skeleton of what the process of using a supervised machine learning algorithm looks like, introducing you to the one that we at MBCR use and have built into our portal, and then we're going to get into a demo of the software used that we use here to make training data for these images and then submit jobs via our portal to achieve segmentations. So just a little bit of background on our group. Where a lot of this is done is a collaboration between two groups called Slash. One group is, of course, here, Chris, myself, Francisco, and a few other people, as well as a group at the University of Utah at the Scientific Computing and Imaging Institute, who are more of the algorithm developers. So they're the machine learning experts who drive the development of these algorithms. And then it's our job to implement them in a logical manner, which is what I'll show here and on the coming slides, and then get them to work on data that are terabytes in size instead of just a small image, which is what most of the community does for algorithm development. So, for example, here in the lower left corner is an outline of a volume, a serial block face volume that I've collected. And I've selected throughout 50 points using the software that I'll show you in a few minutes. And what these points are going to correspond to are where we're going to extract our tiles from that will be used to train the algorithm. So it's obviously, given the huge nature of these data sets, it's preferable to take these points from as big of a distribution of the volume as we can get. If we put them too close to one another, you run the risk of biasing the learning to just that area and not the entire volume. So we typically like to extract tiles from as much of the volume as possible to give it a full representation of that volume. Of course, if you really only wanted to segment that specific area, you would only train it on that specific area. If you only wanted to segment neurons, for example, you could only train it on neurons and say, I don't care about the rest of the data if it looks like garbage, as long as I get my organelles and the neurons looking good. So what we do is we take those points and, using some scripts we've written that you'll be introduced to, we extract about centered at each of those points a tile. Say this tile is 500 by 500 pixels in size. This is what's called a training image. It's just a grayscale image that is going to be fed to the algorithm to teach it what these features look like. Of course, we have to tell it what we care about, otherwise it doesn't know. So this is where the manual segmentation comes in. This is what's called generating training labels. So you can see here, hopefully you can see in that middle image the red shading where I've manually annotated using the software I'll be introducing every instance of mitochondria in that tile. So what this does is then we can make what's called a training label, which is just a binary mask of where all those mitochondria are in that manual segmentation I've made. So the set of both the training images and the training labels are then fed to the machine learning algorithm to tell it where the mitochondria are and where the background are and how to learn what those features look like. So of course this is repeated a number of times to give it enough data to train the pixel classifier to a sufficient level. So say we take those 50 points and do this 50 times. this is really the only step of the process that takes any human time, right? Like I said, for a new user, maybe a day; for an experienced user, a few hours to generate these manual segmentations and then extract these tiles. So as you see, we have a set of training images and a set of training labels, and then those are going to be fed to our algorithm. The algorithm of choice we use here at MBCR is called the Cascaded Hierarchical Model. As I said, this was developed by our collaborators at the University of Utah. The reason we use this is because it's in the community. They do competitions. So every group that develops these sort of algorithms will try to run their algorithm on say a test data set, and then quantify how accurate it is and rank who performed best. So this algorithm is consistently in the top, 1 to 3 algorithms for various data. So it's very cutting edge. It performs very well, especially for the type of data we're working with. So what you see here is the training set in the lower left corner. Again, that's a set of images and manually segmented labels. What happens, just to kind of give you a skeleton, dumbed down sort of schematic of what's happening behind the scenes with the algorithm. It learns the features. It's called feature extraction. So it is then basically what it's doing is it's finding the background and foreground pixels, running these filters on the image, and learning what they look like once these various filters have been applied. From there, it trains a classifier. What the classifier is like I was alluding to earlier, this is what takes that unlabeled image with no prior knowledge and assigns a probability of each pixel belonging to the feature we care about. So you could stop here and be done and say, okay, let's see what we get. The novelty of the Cascaded Hierarchical Model, or CHM, is that using a user-specified number of levels, the images are downsampled. So this is basically reducing, it reduces the size of the image by a factor of 2 each time. So instead of a 500 by 500 pixel tile, we now have a 250 by 250 pixel tile. This is achieved by averaging the intensities of neighboring pixels together and giving you your output pixels. So this is very powerful for EM data because as you downsample, the features look different, right? So if you imagine a really high-resolution nucleus, you can see maybe patterns of chromatin inside of the nucleus that are powerful features to learn at the native resolution. As you downsample it, those features tend to blur together, and that is, again, another feature that can be learned at that scale of resolution that you wouldn't get if you just used one level. So this can be done numerous times. The same features are extracted at each level of the downsampling hierarchy. And then all of the classifiers are combined into one output classifier that operates at the native resolution. This is then repeated a number of times iteratively, so the output is fed in to the beginning and then repeated to refine the classifier. So the whole process looks like this. We feed it our blank training images and our labels. We train the classifier, which again is all done on a machine, so no human interaction. This can take about, depending on the size of your training set, a few hours to a day. And then what we get out are what are called probability maps you see on the right. So that is actually not your final segmentation. So part of the goals of the second session are going to be how can we take this probability map and get a final segmentation, meaning pixels that are just 0 and 1 valued that we can then extract quantitative data from. So you can see with the yellow arrows there, the probability map is pretty representative of where those mitochondria are that it was trained on in this example. So that's all well and good that we can do that, but the question still remains, how can we apply this to huge images, right? So we're up to that point, we were still only using 500 by 500 pixel tiles, but the image is 32,000 by 24,000, so it's still a gigantic problem. How are we going to do this? So one strategy, depending on what feature you're trying to segment, is we can eliminate a lot of the computational cost by pre-downsampling the image. So for big features like a nucleus, represented by that top line there, what that graph is the lateral pixel size. So as you downsample, the pixel size is also decreasing by a factor of 2. So if my native pixel size is, say, 3.5 nanometers per pixel, and I downsample by a factor of 2, it becomes 7 nanometers per pixel, which is that first column of points there on that graph. The F value is a segmentation evaluation metric, which we'll be going over again in the second session. It ranges from 0 to 1, with 1 being a dead-on perfect segmentation, and 0 being just complete trash. So what this shows is a quantification of how well that the segmentation performed at varying levels of downsampled data. And the way we evaluate how well it performed, how we calculate this F value, is by comparing it to some ground truth images. So we run the training, we get our classifier, we get our probability maps, and then on a few of those, we're going to go back and then manually trace every instance of every mitochondrion. And then we can do a pixel-by-pixel comparison, like an overlay, basically, between the segmentation and what's called the ground truth, what we manually segmented. And then we can compute values like the F value to tell us how well the algorithm performed. So what you see here is, depending on what we're interested in, we might have to adapt different strategies to the segmentation process. So, for example, for nuclei, nucleoli, big things, relatively regular texture patterns, there's not a very big drop-off, if any, in quality as we downsample the image. So for features like that, your strategy would be, of course, to downsample the image as much as you can, because if I can downsample this image that's 32K by 24K by a factor of 10 and get it to, say, 3200 by 2400 pixels, that's a gigantic saving in computational time required. For smaller features, more irregular features like mitochondria, lysosomes, smaller membrane-bound organelles, we obviously can't do that, so we have to use the higher resolution data. Downsampling it prior to running the segmentation is not a good approach to take here. So what we do to compute the probability map for the whole image is what's called tiling it. So this has two huge advantages. First of all, we are segmenting a tile that's the same size as what we trained it on. And second of all, this allows for parallelization. So the tiles aren't dependent on, the whole process isn't dependent on any other region of the image. So we can take each one of those tiles, process them separately on different processors, merge them together into one output image. So this is saving a gigantic amount of time. Instead of, say, taking 1 to 6 years to run on a single CPU, we can get an entire data set segmented in maybe 5 to 7 days' time using parallelization across multiple clusters. So here is, for example, the probability map for those nuclei. You can see the correspondence. Nuclei are probably the easiest thing to segment in these data sets. They're very big, they're very regular. We get very good results. The stitching, and then some filtering, which we'll discuss again in the second session to get you down to the segmentation. Yeah.
So it looks like there's some different artifacts then, like roughly nuclear size, sort of full speed. How do you control them?
How are you talking about on the segmentation?
Yeah. No, I mean in the original.
Mm-hmm. Over here?
Yeah. So how do you avoid the segmentation misidentifying those?
So that's something that we'll discuss in the second lecture. So after you get these stacks of segmented images, you can apply a lot of morphological filters. So for example, if you know a nucleus has a certain range of acceptable volumes or acceptable, sphericalness, you can filter out the things that don't meet those criteria. For example, if you look at this final segmentation over here, there are some artifacts like these, for example, right, are probably not. Actually, I think this one is. I think that's one of the capillary endothelial cells. But yeah, so those will drop out really easily once you say, okay, this is way too small or too irregularly shaped to be a nucleus, let's say. Did that answer the question? Okay. And then taking it back to smaller features, of course, like I said, mitochondria, we can't downsample the image as much. So here we've only downsampled by a factor of 2, so going from 32K by 24K to 16K by 12K, just halving each dimension. Obviously, this requires a lot more tiles, as you see here, so this has a much greater computational cost. There's the example output probability map. You'll see a lot more errors here because mitochondria are much more irregular, so you'll get a lot more artifacts. However, like you see, the transition between these two steps, a lot of those drop out by applying the same thing, like I just said, applying morphological filters, applying things that get rid of 3D segmented objects. Again, you see there, for example, here is what I believe is a capillary endothelial cell. That's obviously way too large to possibly be a mitochondrion, so that drops out in our post-processing filter. Then we see the image overlaid. This is just that small little box there overlaid with the results to show what it looks like on the image when we get it out. Okay, so are there any questions about any of that? Yeah.
So, do you always manual segment the highest pixel resolution, or it depends on case by case?
The highest what?
The pixel resolution, for example, like mitochondria, right? You use the downsample by 2, or for nucleus, you do a manual segmentation of downsample by 2.
Well, using iMod, as you probably know, because I know you have experience with iMod, the model files. So what I'm going to show next is how to generate what's called a model file for everyone else using iMod. This is what we use to make our training images and labels. The model files can easily scale between, say you do it on the native resolution, you want it on a bin 10, you can use that exact same model file. You don't have to recreate it to load on the downsampled image. So it depends on a couple of factors, of course. One is how large is the data set. So if you load, depending on the power of the machine you have, a 32K by 24K image and try to segment it, it might be slow. So a lot of times I'll do the segmentation on a downsampled version just because it's faster for my machine. Obviously, you can only do that to the extent of which you can still determine what the structures are. If you downsample too much and you can't tell that it's a mitochondrion, obviously that's a problem. And if you can't tell it's mitochondrion, there's no way the machine's gonna be able to. So basically, yeah, the segmentation can be done on really any resolution you want, and then you can determine your strategy from there. Does that make sense?
Yeah. So it's basically up to me to decide what question am I asking.
Yeah.
Decide what segmentations level I want.
Yeah. So what I usually do when I'm approaching a new project, let's say, that I haven't worked with this type of data before, or this type of, anatomy, is train the classifier at the native resolution. Because like I said, you can transfer that training data over to any resolution pretty easily. I'll then train a classifier at that resolution, run it on just some small tiles, like a 500 by 500 pixel tile. You can then either generate ground truth for that and do a quantitative comparison to see how well it did, or you can just eyeball it. If it's really bad, you'll see immediately. Conversely, if it's really good, you'll see immediately. see how well it did, then maybe rerun it on a binned version. Because at that point, there's no cost to you, really. You've already done the segmentation. At that point, it's just running the job through the portal, see how it did at bin 2, or downsampled by 2. Again, you can quantify it or eyeball it to see. So usually when I'm approaching a new project, I'll do that sort of experiment at the outset to see what I can get away with. And it's worth it because you're talking about, you know, potentially saving many CPU years of computational costs by just running that simple experiment.